everyone, I'm Sheena and welcome to my channel Comprehend Biotech. The topic for discussion today is fluorescent in-situ hybridization, commonly known as FISH. Yes, you heard it right, FISH. But before we start, in case you're here by accident and you have no clue what biotechnology is, please do not leave so soon. Please do check the description of this video. You will find a timestamp that says stay motivated. It usually is at the end of the class that I give out a motivational quote for my students. Maybe it's just the quote that you needed to start your day strong or to end it at a positive note. So please check that out. And for others who are here, the topic of discussion is fish. So let's go fishing. Let's start, okay? Welcome again. So the topic of discussion today is fish. So we'll be seeing how fish is used, or fluorescent in situ hybridization is used to map a genome. Okay, so we'll be seeing fish, the application of fish in mapping. So if you remember in last class on restriction mapping, I had told you that restriction mapping comes under physical mapping, and fish is another type of physical mapping. Basically, in physical mapping, what we do is we find out the distance between certain landmarks. Okay, using those landmarks, we actually map a whole chromosome. So in restriction mapping, we used restriction sites as the landmarks. In fish, we'll be using certain DNA sequences as landmarks. Okay, so also if you would remember, in restriction mapping, we learned about a concept briefly. We learned about optical mapping, right? What did you do there? You directly visualized where the restriction sites were using fluorescence, right? Fluorescence. So the difference over here is, here also you will have a direct visualization of where your DNA sequence is or landmark is, but it is by the technique of hybridization. So we'll be seeing what is fish, what are the steps in it, and how we will use it to map a genome. Okay, so fluorescent in situ hybridization. So what is the basis for molecular hybridization? We all know that the DNA strand, right? What is the structure of DNA? It's a double helical structure. Basically, it is composed of nucleotides. Nucleotides has one nitrogenous base, a sugar and a phosphate group, right? Those three together form one single nucleotide. Like that, many nucleotides join together. They have base pairs, and two strands of DNA together coil up to give the double helix structure, right? So we know that adenine base pairs with thymine with a double hydrogen bond. We know that cytosine base pairs with guanine with a triple hydrogen bond. Okay, so since there are many hydrogen bonds, what happens is DNA is a very stable structure. So that's what is being said over here. Okay, so DNA is basically a very stable structure. And whenever a hydrogen bond between these bases is broken, so if you have two strands of DNA and the hydrogen bonds is broken, it is very easy for the repair to happen. That is the reformation of that hydrogen bond. So that is what is being told over here, that if the hydrogen bonds that hold the helix together are broken with heat or chemicals, the helix is able to reform when conditions become more favorable. So this ability of DNA helix to reform or renature provides the basis for molecular hybridization. So this is exactly what we'll be doing in fish also. We'll be utilizing this particular principle that a DNA, if it has been denatured, it can be again reformed, okay, under favorable conditions. So that is the basis of hybridization. Hybridization is nothing but two different strands coming together, they're pairing with each other, okay. So in situ hybridization. So molecular hybridization could be used to identify the position of DNA sequences in situ. So what does in situ mean? In situ, when we are talking about chromosome, means we'll be able to find the natural position, that is exactly where it is. We are not going to take it out anywhere of the cell or anything. We are going to find out where our particular DNA sequences are on the chromosome. So that is why it is called in situ. Okay, we'll find it at its original position. We are not going to change the position. Okay. So in situ hybridization is a procedure that allows researchers to locate the positions of specific DNA sequences on chromosomes. So what happens in fish? Fish or fluorescent in situ hybridization And specifically in this mapping technique, we are going to use fluorescent probes, okay, to detect the location of DNA sequences on the chromosomes. So as I was telling you, in physical mapping, we use certain landmarks to, totally map the chromosomes. So here in fish, the landmarks are nothing but your certain DNA sequences. So in order to fish those DNA sequences out, we need to have certain probes with us. Now we'll be seeing what probes are and how they are designed in the coming slide. Okay. Now, why fluorescent probes? Initially, you had many techniques using radioactive probes, but then we slowly shifted towards fluorescent probes because they are more stable, they are safer, and they can be easily detected. Okay. So how will we use fish in physical mapping? I hope it is clear by now, right? So in fish, the marker is a DNA sequence that is visualized by hybridization with a fluorescent probe. The position on the chromosome at which the hybridization occurs provides the information about the map location of the DNA sequence used as a probe. So if you have certain landmarks, you can join them together to get a complete route, correct? Just giving reference to the previous topic that we studied of restriction mapping, we found out where and all the restriction sites were there. And using that knowledge and overlapping sequences, you are able to put a framework for the complete chromosome. Likewise, if we know certain DNA sequences and where their location is on the chromosome, using that information, we'll be able to form the complete map of a chromosome. Just like going from place A to B, if you have different landmarks like a school, a hospital, a bank, or a temple, all those landmarks, what will you do? You'll connect each places together and you form a map, right? Just like that, here DNA sequences are your landmarks. What is the fish procedure? We'll see in as much detail as we can. Okay. So the first step, before we start, what do we need? We will be needing a DNA probe and the target sequence. Target sequence is nothing but the DNA sequence which we are going to map. Okay. So that will be our target sequence, and we need a probe. So the first step is probe design. So what is a probe, right? So probe is nothing but a single-stranded DNA or an RNA piece, okay? And that will be designed by us in such a way that it will be complementary to our sequence of interest. Whatever sequence we are targeting, it will be complementary to that. So probe is something that we design, okay? And remember, it has to be a single-stranded molecule. Only then it will go and hybridize with a single strand of our target sequence, okay? So before hybridization, the DNA probe that we have designed is labeled by various means such as nick translation, random primed labeling, and PCR. In this diagram, you can see that we are going to design a probe which is complementary to a gene. Okay, so generally we use fish to actually map out a chromosome using where is the position of a particular gene located. Okay, so you can say the DNA sequence that we are going to find out is nothing but a gene of interest. Okay, so you're going to design your probe, so it has to be complementary to the gene. So you have to take that gene of interest. So please understand, you know the sequence of the gene, but you're not aware where the position of the gene is there. So that is why you're going to use the technique of FISH. So wherever your fluorescently labeled probe will go and hybridize, it will give us a location of that particular gene. Okay, so you take the known gene and we're going to add DNAs to it. On addition of DNAs, it will create certain nicks in the DNA strand. So you can see over here, right? It is creating certain nicks over here. That means it is removing nucleotides from here. In the next step, depending on whether you want an RNA probe or do you want a DNA probe, you will be adding labeled nucleotides. So here in this example, they have added DUTP, which is digoxygen, and it has a digoxygen molecule attached to it, or a biotinylated DUTP. So it could be a biotinylated ATP also, or DATP or DTTP, anything. That is according to how we are going to design our probe. We just need it to be labeled. Now, there can be two types of labeling. So I'll just show that to you. But nick translation is basically this. Wherever this nucleotide was removed, you'll be adding a labeled or a molecule which has been added with the hapten. Okay, DNTP added with the hapten. And I'll show you one second. Yeah, so there can be two ways of labeling the probe. You can have indirect labeling or you can have direct labeling. So what happens in that? So for indirect labeling, probes are labeled with modified nucleotides that contain a hapten, whereas direct labeling uses nucleotides that have been directly modified to contain a fluorophore. So I'll explain with the diagram. So over here you can see, right, this is direct labeling. So the nucleotide, whichever you added, right, it is already having a fluorophore to it. So you don't have to do, again, addition of fluorescent molecules. You don't have to do in the next stages, okay. Already in the beginning, after Nick has been formed, you are adding a fluorescently labeled nucleotide. That is direct labeling. Another type, the previous diagram that we saw, was indirect labeling. That means you add just this portion over here that you see, right? You will be adding a nucleotide with a hapten. Now, hapten could be a biotin molecule. Basically, what we'll do is, after hybridization, we'll add a fluorophore, which will go and attach to this hapten molecule, and then it will be detected. So that is kind of indirect detection. Okay, so you can see over here, right? So that happens after hybridization. Okay. So that was probe design. This is all before the hybridization step. I just explained to you what are probes, how are probe designed, and there are two types of probes that you can have based on the labeling that is done. It could be direct labeling and indirect labeling. Okay. So step 2 is denaturation. Okay, so the labeled probe and the target DNA are denatured. So if this is your probe, it is labeled. Okay, you can see over here, right? If you remember, this is the example of indirect labeling. This is the example of direct labeling, because this dot over here shows the fluorophore. This this is representing nothing but a hapten that has been attached, and later a fluorophore will be added to it after hybridization. Okay, so you have your probes which are ready. You have your target DNA sequence. We do not know where the gene is. We'll be finding out the location of the gene. So you have the target DNA. You're going to denature both of them. Okay, so here you can see both of them are to be denatured. Now, the standard method for denaturing chromosomal DNA without destroying the morphology of the chromosome is to dry the preparation onto a glass microscope slide and then treat with formamide. I'm sorry. So what you're going to do is, we want the DNA to get denatured, but we do not want the shape of the chromosome to be denatured as well. So we do not want to disturb the shape of chromosome. So the safest way to do that is you're going to put the chromosomal DNA. First of all, you're going to select cells which are at the metaphase stage, okay? Because at that time, your chromosome is very tightly bound, and it has a perfect shape, or most tightly bound shape, most defined shape during that metaphase stage. So you will select cells that are in the metaphase stage, and you will fix those chromosomes onto glass slide. You'll dry it up, and then you will treat it with formamide. So what happens is, the chromosome is not destroyed. The shape of the chromosome is not destroyed, but internally the DNA is denatured. Okay, the double strand becomes single strand. Okay, the basic shape remains as such. That is what is being shown over here. Dividing cells with metaphase chromosomes are fixed on the microscopic slide. You treat it with formamide, so the chromosome becomes denatured. Okay, that is just shown with a representation over here. So the next step after denaturation, your probe is also being denatured. Your target DNA sequence has also been denatured. Next step will be hybridization. Okay. So combining the denatured probe and the target allows the annealing of complementary DNA sequences. That is, hybridization takes place. The fluorescently labeled DNA, so this could be fluorescently labeled before, or if a hapten containing probe. So don't get confused, okay? So the fluorescently labeled DNA finds its matching segment on one of the chromosomes where it goes and sticks. So you can see over here, right? Wherever your target DNA is there. So this is a long chromosomal molecule, okay? And wherever your probe finds its complementary piece, it will go and stick over there. So after hybridization, what do? We have to visualize it. So by looking at the chromosomes under a microscope, a researcher can find the region where the DNA is bound because of the fluorescent dye that is attached to it. This information does reveal the location of that piece of DNA in the starting genome. So for example, after the hybridization has been done, if you observe this microscopic slide under a fluorescent microscope, you will see the fluorescence wherever your target DNA or target DNA has been hybridized with the fluorescent probe. So you can see over here, signal from the probe. This is where your probe has gone and attached. Okay. If the probe has been labeled indirectly, that means initially there was only a molecule of hapten attached to the nucleotide, what will we do? An extra step is required for visualization. So after hybridization, you have to do an extra step. What is that? So we have to do the visualization of the non-fluorescent hapten that uses an enzymatic or immunological detection system. So basically you can add antibodies with anti-dig, whichever hapten you're using over here. If it is a digoxygenin, you will use anti-dig. If it was a biotin, you will use avidin, because biotin has a strong affinity towards streptavidin as well as avidin. Okay, so that is what they're saying over here. They'll use that, and this fluorophore will go and attach to specifically to this hapten containing molecule, right? And then you can carry out the visualization, right? So direct versus indirect labeling, what is the use of both, right? So whereas fish is faster with directly labeled probes, you do not have that extra step, right? Hybridization, after hybridization, you just need to visualize. Even though that is faster, indirect labeling offers the advantage of signal amplification by using several layers of antibodies, and it might therefore produce a signal that is brighter compared with background levels. So you can have this, you're using different antibodies, right? You can use one top of the other. You can amplify the fluorescent signal that is coming out. So it is more easily visualized. Okay, so if one is faster, the other one is more visually or accurate. We can identify it in a much better way. Okay, so that is direct versus indirect labeling, the advantages of both. Here is a diagram that shows the fish in a nutshell. Okay, so you have your chromosome, it has been denatured. You have your probe DNA, which has also been labeled with a fluorescent dye, and it has also been denatured. After denaturation, both of them will hybridize, and then you can visualize it under a microscope, and you will see wherever the probe has gone and attached itself, that is where your DNA sequence or your gene of interest is present. So this illustration over here, it shows how fish can be used to produce a genetic map. So the photograph on the left, that is this one, it shows chromosome number 17 from 4 British peppered moths with fluorescent probes indicating the physical position of specific genes. So this is the physical position. You can see different colored fluorescence over here, right? So they all represent different genes, okay? The illustration on the right over here, it shows the relative positions of the gene on the chromosome. So this is how the mapping is done. So this particular gene is over here, this particular gene is over here, and the distance between each will be known. And thus you can map the whole chromosome. Okay. So as an end note, historically, fish results played a primary role in mapping genes on human chromosomes. Results from these experiments were collected and compiled in databases, and this information proved useful during the annotation phase of the Human Genome Project. So basically, it was a big tool used during the Human Genome Project. But now Human Genome Project, everything is over. What is the current use of fish? So currently, human fish applications are principally directed towards clinical diagnosis. So basically to understand where exactly the disease-causing gene is there on the chromosome. So to do comparative studies like that, we use fish technique right now. Okay, that was fluorescent in situ hybridization. So we saw how that can be applied in mapping or physical mapping of a genome, right? If you have any doubts, please do ask me in the comment section, okay? And as always, I will leave you with a quote. The one for today is, "Peace begins with a smile." It's a quote by Mother Teresa, a very simple one, but a very powerful one. We do not generally realize the power of simple smile, but you never know what ice is being melted by the warmth of your smile. Imagine you're entering a room with people who are having certain kind of hostile feeling towards you. Just enter with a smile and you will see that initial tension just melts away. And I'm not talking about just any kind of smile. I'm talking about a smile that when you look at the other person in the eye and you smile, and the twinkle in your eyes actually lights up the whole world of the person standing right in front of you. You have no clue the kind of power a smile has. And how do we have such a kind of smile? That smile can be within us or can come from us only if we are peaceful inside. When we know how grateful we should be for the things we have and not worry about the things we do not have yet. So smile. Let that bring peace to you as well as to the one who is standing in front of you. You never know what changes it can bring in the other person's life. I hope this video brought a smile on your face. If it did, please do like, share and subscribe to my channel Comprehend Biotech. Until next time.